right. Welcome to my presentation. My project is named Utilization of Woody Biomass and Residues for Production of Energy Products and High-Value Natural Compounds, a case study using biomass from coastal forestry resources. My supervisors are Professor Tanja Barth from the Department of Chemistry, Inger Elisabeth Moran from the Department of Biosciences, and Camilla Løhre, again from the Department of Chemistry. This is a multidisciplinary project, so the focus will be somewhere in between chemistry and biosciences, with tilting more towards chemistry since I am a chemist. So fossil fuels, as we all know, is a finite resource. The rate at which this resource is emptying is, of course, for debate. We are highly dependent on fuels for energy and materials derived from this finite resource. It is therefore desirable to find alternative solutions which are renewable. One of these solutions is utilizing biomass, and is what I'm primarily going to talk about here today. Whether fuels on biomass produce less or the same amount of CO2 is debatable, but that CO2 will become part of the carbon cycle, where biomass in forms of trees and plants stores the CO2 produced, going in a loop. The biomass is in between, the biofuels is produced between here. The combustion of biofuels will emit CO2 into the atmosphere, and that same CO2 will be consumed by other biomass going in a loop. Unlike with fossil fuels, where basically a lot of carbon which is stored already in the reservoirs get pumped up and burned into the atmosphere, not going back, not naturally at least. As I think with every new solution for renewable energy, biofuels is not the sole solution, but part of it. Vehicles on batteries are becoming more standard, for example. So the need for fuels, at least for personal cars, will not increase a lot. But bigger vehicles, such as ships and planes, still need fuel. And maybe even more importantly, a lot of materials are derived from petrol. The production of these materials must, of course, have a feedstock. Battery is not enough to solve that problem. These products include plastics, food additives, medicine, cosmetics, platform chemicals, etc. So in terms of biofuels, we tend to categorize them into first and second generation, and nowadays even a third generation actually, but I'm just going to focus on first and second. First generation biofuels are definitely the more established process, utilizing edible crops to produce, for example, bioethanol and biodiesel. The downside of doing this is exactly that these crops are edible. We're then using a potential food source. So is it right to make biofuels out of food instead of feeding people? Second generation biofuels are derived from non-edible plants. This way we do not compete with food sources. If we utilize forests, we don't even compete with farmlands. We can then use land areas which is already currently not being used for anything. We can even use residues from existing agriculture. Like we do not eat the whole plant from agriculture, we only eat the fruit or whatever plant it is. If you think of deforestation when I talk about utilizing forests, that's understandable. I think it is important that countries with a sustainable forest growth use their forests, like Norway, for example. And even better would be to use residues from already existing forestry. Now we have arrived at the focus points of my PhD project, namely using residues from forestry in Western Norway. So the non-edible biomass I talked about is called lignocellulosic biomass. Take trees, for example, which comprise of mainly cellulose, which is glucose bonded together in a chain, hemicellulose, which are more of a branched structure, which contains more different sugars, and then lignin, which is a formless aromatic molecule, which I'm going to talk about further later. In addition, it's a few percent of ash, wax, fats, and protein. All these constituents will be important further down the line when I talk about conversion to biofuels. So in Norway, there are 978 million cubic meters of forests, and Norway has experienced an increase of forest by 16.2% since 2010. So that is the sustainable forest growth that I was talking about. UNESCO has established a biosphere program. Biosphere reserves are defined as learning places for sustainable development. They are sites for testing interdisciplinary approaches to understanding and managing changes and interactions between social and ecological systems, including conflict prevention and management of biodiversity. My research takes place in one of these reserves, which is Nordhordland. Nordhordland is part of this program and has been defined as one of these biosphere reserves. Within Nordhordland, there are a lot of forestry going on, fortunately for me, particularly Sitka spruce. So this is an interesting species to study. There's also a lot of pine and birch. Juniper is also interesting. This is a mostly unwanted plant, which occupies a lot of space and has no value forestry-wise. So this is the Biosphere Reserve. This is north of Bergen, for those who are not familiar. This is mostly Nordhordland and the northern parts of Øygarden and Faya as well. So to be a little bit more precise, in my project, I'm focusing on residues from forestry. I try currently to map the chemical variation within and between different tree species, particularly spruce, pine, and birch. In addition to residues, forestry industries seems interested to bring value to what is viewed as low quality wood. And that is, for example, old trees, which they cannot use. If what is viewed as low quality wood is equally fit for conversion of biofuels, everyone wins. So to convert biomass into second generation biofuels is not just one process. There are multiple process methods that yields different kinds of products, everyone with their pros and cons. The method for conversion I am focusing on in my project is hydrothermal liquefaction. The advantage of this conversion method is that no drying of the biomass is required. The drying process would take time and energy to perform, so it is a desirable process step to skip. The lignin to liquid method developed here at UIB uses formic acid as a catalyst. This method was intended to convert lignin specifically, which was one of the constituents I mentioned. But this same method proves efficient when converting the whole biomass as well. The reasoning for focusing so much on lignin is that cellulose, which is one of the main components, has been very desirable to make paper. So in the paper industry, they extract the cellulose and the lignin becomes a waste product. If this waste product could be used to make biofuels, its value would increase. So biomass, a reaction medium, in my case water, and a catalyst is exposed to temperatures of 350 to 380 degrees Celsius and high pressure in a sealed reactor over several hours. This in turn yields bio-oil, water, a solid phase called coke, and a gas phase. The oil phase will resemble crude oil from the oil reservoirs, being viscous, often a dark substance. So this is the reactor we use in lab scale. It can contain a few milliliters. And this is basically the same thing, just a whole lot larger. So this we use for large scale testing and contain a few liters. So lignin, the challenge with lignin is that it's not just one particular molecule. It's these 3 units can be bonded together in different arrangement and form this kind of molecule. Some lignocellulosic biomass has lignin that is mostly comprised of one type of unit, while others can be comprised of one of the others. The biofuel produced by lignin or by biomass containing lignin will therefore vary. This is one of the main challenges of utilizing second generation biofuels versus just fossil crude oil. There has been some research on how the temperature, concentration of catalyst, and time affect the bio-oil yield in this process. But what I am interested in is how the composition of the feedstock affects the yield and the composition of the bio-oil produced. The first step in this process is to map the composition of the feedstock. In my case, I chose 3 different species of trees. This mapping process takes time and resources. So to streamline this process, I am making a calibration model based on infrared and or near infrared spectroscopy. These analysis methods require next to no time at all to perform, does not destroy our sample. If enough infrared or near infrared specters are performed, we can use them to make prediction of the composition in an unknown sample. This will make the basis of my first article, hopefully. I will try to compare infrared and near infrared to see which method gives the best prediction power. So to make this calibration model, I have to cover a larger variability. So I divided samples from 3 different trees, spruce, birch, and pine, growing naturally on the coast of Western Norway. These are fractionated into heartwood, bark, twigs, and leaves or needles, yielding a total of 36 different samples. This will contribute to variability in the model, since, for example, bark contains more lignin and heartwood is mainly cellulose. The sample set can thus also be a source for further elucidation of the variability of chemical composition within and between different species of different parts of the tree. So this is the flow chart of the calibration model I am making. We start with biomass and we have to oven dry it so that water don't interfere. We also have to reduce the size of the particles, making it look like kind of sawdust. Then we can take our spectra. So I chose infrared and near infrared to compare them and then pre-treat them, and then they are ready. So with the biomass, we have to decompose it to characterize the different constituents. I'm not going very much into detail about this. This is the procedure I do in the lab to map the constituents. I also extracted essential oils to determine how much extractives there are in my samples. By doing this, I have determined the ratio of lignin, carbohydrates, extractives, and ash in all my samples. The spectra are then used as dependent variables to predict the different ratios of constituents in the biomass. No previous studies have compared data from different spectroscopic techniques to evaluate how to get the best prediction power. So this is an example of how the infrared spectra look like. This is one spectra from all the 36 different samples. As you can see here, all the spectra are different and will contribute to the model. So I use these spectra to predict the ratios. So further work will include my developed calibration method to compare how different compositions in the feedstock affect the yield and composition after the hydrothermal liquefaction. This includes mapping the constituents beforehand and then do the hydrothermal liquefaction and see how that is correlated. I'm also interested to review the hydrothermal liquefaction method used on the whole biomass versus, for example, extract the cellulose beforehand and then use the hydrothermal liquefaction on the residues. The cellulose I can then use to, for example, ferment it to make bioethanol. I will also look into the value of needles. There are quite a lot of value in essential oils extracted from, for example, spruce needles. And again, I could use the hydrothermal liquefaction method on what I have left from this process. So yeah, that's me.